Hey Bio-3 students, this is actually my third try. First try, you guys let me know that the audio wasn't working. No idea why, my microphone just stopped working. Second try, the slides wouldn't advance. So this is my third try. So this is my reminder to be patient with you as you navigate this technology in a format that none of us would really prefer to be doing, and to just stick with it to get it done and to start maybe a little earlier. So this lecture is really just to introduce you to how to go through your lectures for this course. You should take notes. There's a lot of pedagogical evidence that says writing down as you hear the material is a great way to help you remember it. It helps you remember where you wrote it down, how to go back and look at things. Pause the video so that you can write down any questions you have, or note what you don't understand, or make a comment that you would like to bring up in the discussion. Because unlike a face-to-face class, you have the opportunity to pause this video whenever you need to, and to rewind and watch it again if that's necessary. Okay, the slides are advancing. So a nice little image of the Earth, the biosphere, and here we've got some images of blue-green algae, which is more appropriately called cyanobacteria because they are prokaryotic. We'll get into the different kinds of cells in a moment. Under a microscope, they appear with these green areas in them because they are photosynthetic, and they can, where there's intertidal zones, accumulate and make these large structures called stromatolites. This is an image of E. coli. In this class, we are going to be working primarily with eukaryotic organisms, bigger cells that have a nucleus. E. coli are bacteria, and that means they do not have a nucleus. They are cells, and they do contain DNA, but they don't have a separate compartment that their DNA goes in. E. coli is important to human health. We have them in our intestines, like the slide says. They help us to absorb vitamin K, but if we ingest them where they were present in a different animal's digestive tract, they can cause very serious illnesses in us, and that happens because we might ingest undercooked meat. If the meat is cooked well, then it's usually not a problem. Nice little slide showing us the intersectionality of different fields of study with biology. In this class, you all had to survive chemistry to be here, and that is because cell biology is really the cellular chemistry. We have to know how the molecules build into the structures and how those chemicals interact in the cells. We have Sir Francis Bacon. His name, in terms of this class, not as important, but he was the first to define and codify the scientific method, how we go about being very logical and methodical in recording data and being able to demonstrate how natural phenomena work. So our scientific method does depend on us coming up with a hypothesis. And a hypothesis is an educated guess. We make observations. We ask a question about how we think something works. And then once we have that testable hypothesis, and it has to be testable, there has to be some way to measure what you think is going to happen. Make that prediction and do the experiment. Of course, we analyze the results, and then either the hypothesis is correct and we report the results, or the hypothesis is incorrect and we try again. Either way, even if our hypothesis is correct, we do still have to repeat the experiment. Doing it once is not enough to prove that that's the way it really works. And different types of reasoning we can do would be inductive, where we make a lot of specific observations to draw a general conclusion, or we can start with some sort of very general idea and make specific predictions. Both of these are very valuable depending on the types of questions we are asking. A cute little picture of a baby squirrel. Scientists use the knowledge they had from previous behavioral studies to be able to know how to reunite animals that have been separated due to natural disasters. And the Human Genome Project, right? Chemistry, biology, physics, ethics, engineering, all of those had to come together for us to be able to extract human DNA be able to determine the sequence of the particles in that DNA, and those are what will make up the genes, which are the specific regions in the DNA that encode for particular traits or characteristics. Now, of course, it's not the DNA sequences of every single person on the planet, but it is a comprehensive library of all of the genes that are in human beings. And we hope to be able to use that information to know the specific causes of genetic illnesses and potentially be able to treat them. Went the wrong way. There we go. Cute little toad, highly organized, and animals start out with molecules. Those molecules build into organelles and cells. The cells build to tissue, tissues to organs, and organs to organ systems. Animals, plants, very complex. But there are single-celled organisms that also have their complex organization. They just don't get above the single-celled level. The sensitive plant is a plant that has the ability to move when you touch it. It won't get up and walk away, but its leaves will fold. And that causes a reaction that's immediate, and then eventually the plant is able to open up its leaves again. And that takes a lot of energy for a plant to be able to do that. And when we get into our chapters on metabolism, we'll talk about how those energy transformations can take place. Our little kitty is a nice example of genetics. The mother cat has 3 kittens. The kittens are all distinct from each other. And none of the kittens has the same fur color as its mother. So we're going to talk about genetics. We will use animal, plant, bacterial, and human genetics as examples. And we'll learn how that information in our DNA has discrete entities called the genes, particular regions of that DNA, and how those pieces of DNA are inherited and recombined between parents to create offspring that are new combinations that didn't exist in either parent. Our lovely polar bear, example of adaptation. Now the polar bears weren't created because the ice was white. The animals that live in Arctic and Antarctic regions survive and compete better because their coat or feather colors camouflage them. Predators can hide and sneak up on their prey, and prey can hide and be uneaten by their predators. Our nice California condor being used here as an example of energy conversions. We're going to talk about metabolism at the cellular level, but that can be built up to look at metabolism at the organism level. So the energy that condors and we get from our food gets converted into cellular energy to do work for us. This is a DNA model. It's late for me. Each of those little dots represents an actual atom. You learned in your chemistry class that atoms combine because the electrons in their valence shells can interact. Depending on the number of valence electrons will determine the number of bonds an individual atom can make. DNA is an organic molecule, and in terms of a biochemistry class, organic means that it is carbon-based and that it is made by living things. So DNA has a backbone of carbon atoms, and the atoms that are attached to that are hydrogens, oxygens, nitrogens, and phosphorus. And they join in a very particular order to create this very organized, very large molecule. Now the levels of organization, like we saw in our toad, we have molecules. Those molecules will make organelles. In eukaryotes, those organelles have their own individual membranes, so they are individual compartments. In prokaryotes, they don't have a nucleus, and they don't have any other internal compartments that have their own membrane. But if we're looking at something that is eukaryotic, those organelles will combine and coalesce and be surrounded by a cell membrane to make cells. Cells of the same type combine to make tissues. Tissues combine to make organs. different organs make organ systems. For example, our digestive system. Our tongue is an organ, our esophagus is an organ, our stomach is an organ, our intestines are organs, our spleen, our liver, our gallbladder, and all of those make up the system of those interacting and interdependent organs. Organisms, all the different types of species that are in a particular area or on our planet. Populations are a group of individuals of the same species that have the potential to interbreed. And communities are groupings of populations. By the time we get to the level of an ecosystem, we're talking about all the living and non-living things in a particular geographic area. And then we have the biosphere, the entire planet. This phylogenetic tree of life, we have a "you are here" star. We're out here on the branch that says eukarya. And there are two other branches, the bacteria and the archaea. That means the ancient ones. Now, a phylogenetic tree, the closer two branches are to each other means the closer they are evolutionarily, which means the closer they are in geologic time. So when we look at this, there is the one branch point, which is the common ancestor to all living things. We have the bacteria domain going off this direction. The next one that branches off from that universal ancestor is the archaea, and then the eukarya. So we can interpret this to mean that the eukarya domain is more closely related to the archaea domain than it is to the bacteria, but the archaea are closer to the bacteria than they are to the eukarya. And we look at us, we are very closely related to plants and fungi. Ciliates are single-celled protists. They have little tiny hairy projections that make them move. We're also pretty close to slime molds. Now this tree was constructed by Dr. Carl Woese, and he used ribosomal RNA genes to be able to create these groupings. Now, RNA stands for ribonucleic acid. It is related to DNA. DNA stands for deoxyribonucleic acid. And ribosomes are a structure composed entirely of RNA and protein. And there's two components that kind of make a sandwich. And those ribosomes, their function is to read RNA and generate proteins. And just about every function in our cells, in our bodies, depends on the activity of proteins. So this is a very, very basic function in all living things. So how proteins are made is very similar. So the ribosomal RNA stays conserved among extremely related organisms. So once you get to a certain number of differences, it puts the organism in a different domain. So really the only difference is types of ribosomal RNA. All of these organisms have it. And the bacteria and the archaea, both of these domains are entirely single-celled organisms. Nothing with more than one cell. And they also do not have a nucleus. That means they are prokaryotic. They have a cell membrane. They have DNA. Most of them have a cell wall, but not all do. But they don't have any type of internal compartment. The eukarya are all organisms that do have internal compartments. So they have a membrane around their DNA, and they have lots of other independent compartments inside of their cells. So some images from the domains. We've got bacteria here, and these are the archaea. It says extremophiles. This would be a picture from Yellowstone. So this is around a geyser. So the temperatures are extremely high. The water is often of a very low acidic pH. And the archaea are hypothesized to be very similar to the first organisms that were living on Earth under similar, very harsh conditions. We've got a plant and a lion. Plants and animals are both eukaryotes, or domain eukarya, because their individual cells have a nucleus. We have an image here of a scientist extracting DNA. He is wearing a mask and gloves because he is protecting the samples from him. We do not want any DNA from the scientist ending up, especially in a criminal investigation lab. Now, in a face-to-face section of this course, we would be extracting our own cheek cell DNA and doing some experiments, being able to visualize that. Won't be able to do that because it requires tools to make multiple copies of that DNA in order to be able to see it. But what we will be able to do later in the semester is to extract DNA from plants. So we can do it from wheat germ, we can do it from strawberries. There's lots of different ways that we can get DNA and at least go through that procedure. And lastly, we have some researchers excavating dinosaur fossils. Why researchers doing dinosaur fossils when this is not an anthropology class or an archaeology class? Because fossil evidence ties us to our place in evolutionary history. If amber is found, they often have organisms trapped in there, and DNA has been extracted from those ancient preserved specimens. And we will get later in chemistry talking about carbon dating and evolution and the types of evidence for that. So that wraps up this very brief introductory video. Remember that you should have been taking notes. If you didn't this time, that's okay. Do it next time. But you do need to now go to part 2 of the discussion, which is to post some manner of thoughtful question or a comment. There are instructions for that in the discussion, and that's worth 2 points, and it has to be done by 11:59 a.m. on Monday, August 24th. And then you will need to come back after you post something. You can see what other people post, and you need to reply to at least 2 classmates. And it can't just be yes, I agree, or ooh, that's cool. Some sort of comment that adds to the discussion, because if we're in face-to-face class, we would be having discussions with each other and exchanging ideas. You have till 11:59 p.m. on August 24th to do that part. You also have the labs to do. A couple of people have already gone in there and started playing with it. If you have technical issues, I have opened up a chat. It'd be great if people can post in there, because we might be able to troubleshoot for each other, or if we see that we're just not able to fix it, then I can send a request to Lab Archives to get help for everyone and try to make this less frustrating. Once we get a rhythm going in the first couple of weeks, it will be a lot smoother. I know you're all dedicated, bright little scientists. We're going to make it through this, and we will do so with patience and compassion and as many smiles as we can manage.